हेलो स्टूडेंट्स, वेलकम बैक टू योर फेवरेट चैनल। हम आज पढ़ेंगे योर इंटरेस्टिंग टॉपिक माइक्रोबियल ग्रोथ। सो व्हाट डू यू मीन बाय द ग्रोथ? Growth can be defined as increase in all cell constituent resulting in increasing mass and size of cells. That means if the cell size is small and gradually the cell size is large, that is also called growth. And the cell number also increase, that is called growth. So what is microbial growth? In case of microbes, the cell number if increase, that is called microbial growth. Generally microbes divide by binary fission, fragmentation and budding. Binary fission is the most common method of reproduction in bacteria. It is a symmetrical process. That means the parent cells equally divided into two daughter cells. The microbial population double at a specific time interval. That interval is called generation time. So in case of the E. coli bacteria, the generation time is 20 minutes. The process of binary fission: first the bacterial DNA get doubled, that is the process by DNA replication. Then cell wall in cell wall constriction form. After constriction formation, septum will be formed which separate the daughter cell. Finally the daughter cell separated. So the separation of daughter cell can be done by mesosome. The mesosome is mesosome. It is the plasma membrane folding, the folding version of the plasma membrane, that is called mesosome, which help in DNA replication and daughter cell separations. So second process is fragmentation. In this process, the body of the bacteria or microbes gets some fragmented due to some accidental breakage. So this method is also known as splitting method of reproduction. Mostly microbes like fungi use this process of reproduction. Next हम पढ़ेंगे budding. Budding means it is a one cell bud is formed from the parent body. It is new bud. It is parent body. It is asymmetrical process because the parent is not equally similar to in size with the daughter cell. So that is asymmetrical method. Next हम पढ़ेंगे microbial growth types on the basis of nutrition in batch culture. So what is batch culture? What is batch culture? We use growth media for microbes, that can be solid culture, that can be liquid media. In batch culture, only one time nutrients give to culture media. that is called batch culture. Different type of cultures are there, that are batch culture, continuous culture, synchronous culture. In batch culture only nutrients are available to the bacteria for once. In continuous culture, continuous mannerly we use nutrient media. What is monozygotic growth? Monozygotic. Mono means single time. Oxic means oxic. That means nutrients. The growth of microorganism can be studied by help of batch culture with one time supply of nutrition. That is monozygotic growth. The growth of microorganism in batch culture show four phases, that can be lag phase, log phase, stationary phase, death phase. In a diagram, A represent lag phase, B represent log phase, C represent stationary phase, D represent death phase. What is lag phase? In this phase, the bacteria get physically active and adapt the external environment. Before dividing, the microbes first adapt the external environment and physiologically mostly active. There will be no increase in the number of cell, only physically active and adapt the environments. The time of lag phase depend of the state of bacteria or type of bacteria. State mean young or adult, old, like. Log phase. In this phase, the bacterial grow, and the phase is also called exponential phase. In this phase, cells started to divide and metabolical activities at its peak. So that is called exponential phase. The total number of cell at any given time can be calculated by the formula Nt is equal to N naught to N. Nt is equal to total number of cell at a time t, or after time t number of n0. N naught number of cell at time 0. 2 to the power n is total number of cells, n is generation number. For the example, calculate number of cell t is 60 minute, n naught is equal to 10, for E. coli. For E. coli divide in every 20 minutes. E. coli divide in every 20 minutes. So number of generation n is total time, that is 60 minute, and the E. coli use to divide two cell require time, that is 20 minutes. So 3 generation. 2 to the power n is equal to 8. So nt is equal to n naught into 2 to the power n. N naught is 10 and 2 to the power n is 8. So answer is 8. Next, stationary phase. In this phase, the bacteria cannot continue to grow, or microorganisms cannot continue to grow. So this phase is called fixed phase. Why the bacteria cannot grow? Because of the essential nutrients will get finished, or accumulation of toxic metabolites, or critical population may be achieved. So in this phase, number of cell increase is equal to number of cell decrease. Next, last phase is declination phase or death phase. It is also called death phase of microorganisms. Why the microbes death? Because of the all the nutrients media get finished. So there will be the decrease the two causes. There one cause is decrease of the accumulation of excess toxic. The number of cell get decreased because accumulation of toxic matter and nutrients are finished. Next, diauxic growth. So diauxic growth is also called diphasic growth. Diauxic growth. In diauxic growth, the bacteria's growth in two phases, and the growth of microbes occur in two types of nutrition. That means the microbes uses two type of substrate like glucose and lactose. Diauxic growth can be observed during the cultivation of E. coli material cell containing both glucose and lactose. At the time of culturing, first the microorganism uses glucose. Next, after finishing the glucose, it starts utilizing lactose in the second phase. In diauxic growth, second phase is small than first lag phase. Hence here is a growth curve which represents the growth of bacteria. हम देखेंगे the growth curve of the diauxic growth. A is the first lag phase. In this phase, the microorganisms actively physiologically get activated and adapt to the external environment. Then in the B phase, that is first log phase, which is greater than the second log phase. Okay, so second is short lag phase, that is C. C is the first stationary phase and D is second short lag phase. In this short lag phase, the microbes use lactose, and E is the second log phase, and F is the stationary phase, and G is also there will be G. G is a declination phase or death phase. So it is very important for you for your examination. हम नेक्स्ट क्लास में पढ़ेंगे वायरस.